All right, everyone. So now we're diving into a specific piece of research that shows you how genetic studies actually get done in real life. This is the Oruc study from 1997, and it's a perfect example of what we call association analysis in psychiatric genetics. So we've been talking about how genes might influence depression and bipolar disorder, right? But how do researchers actually figure this stuff out? How do they go from maybe genes are involved to actually finding specific genes that might matter? That's what this study is all about. So let's start with what you need to learn from this study. So by the end of this lesson, you should be able to do some key things that will help you understand how genetic research actually works in practice. First, you'll be able to explain association analysis in genetics. This is the method that researchers use to figure out if certain gene variants are linked to mental health conditions. It's like the detective work of genetics. Think of it as comparing fingerprints at a crime scene. You're looking for patterns that might connect the evidence. Second, you'll describe Oruc and colleagues' aims, methods, and findings. We're going through this study step by step, so you understand exactly what they did and what they found. Third, you'll evaluate the study's strengths and weaknesses, because no study is perfect, right? You need to think critically about what this research can and can't tell us. And finally, you'll understand its scientific and clinical significance. Why does this study matter? What does it contribute to our understanding of bipolar disorder? Even when studies don't find what they expect, they can still teach us important things. So let's start with the basic method. What exactly is association analysis? Association analysis is actually a pretty straightforward concept when you think about it. It's a method to compare genetic variants in people with versus without a condition. So you take a group of people who have bipolar disorder and a group who don't, and you compare their genes to see if there are any differences. Think of it like comparing the contents of two different toolboxes to see if one has more broken tools than the other. Think of it like comparing umbrella use in wet versus dry people during rain. If you notice that wet people were more likely to have broken umbrellas, you might think maybe broken umbrellas make you more likely to get wet. It's the same logic with genes. You're looking for patterns that might explain differences between groups. In this study, they were looking at the relationship between specific gene variants and bipolar disorder. They wanted to see if people with bipolar disorder were more likely to have certain versions of genes compared to people without the condition. But why did they choose to study these particular genes? The researchers didn't just pick random genes to study. They had a really good reason for focusing on serotonin-related genes. Their focus was on serotonin-related genes. Remember from our previous lessons how serotonin is involved in mood regulation? Well, if serotonin problems contribute to mood disorders, then genes that control serotonin might be good candidates to study. It's like investigating the electrical system when your house lights keep flickering. One gene they looked at was 5-HT2C, which is a receptor linked to appetite and mood. This receptor sits on neurons and responds to serotonin, so variations in this gene might affect how well the receptor works. The other was 5-HTT, which codes for the transporter that clears serotonin from the synapse. Remember those little vacuum cleaners we talked about? This gene determines how well they work. These genes were chosen based on prior biochemical research. It wasn't random. They had theoretical reason to think these genes might be involved in mood disorders. So with that background, let's look at what exactly they were trying to find out. Let me break down their approach for you. Their aim was to find if specific gene polymorphisms are linked to bipolar disorder. A polymorphism is just a fancy way of saying different versions of the same gene, like how some people have blue eyes and others have brown eyes. They wanted to know if certain gene versions were more common in people with bipolar disorder. This was a correlation study, not an experiment. They weren't manipulating anything or giving people treatments. They were just looking at naturally occurring differences between groups, like comparing the height of basketball players versus the general population. You're not making anyone taller, just measuring what's already there. You can refer to the table if you want to remind yourself of the differences. Reminder, Correlational research identifies relationships between variables. They compared alleles frequency in a bipolar group versus a control group. An allele is just one version of a gene, so they were asking, do people with bipolar disorder have different versions of these genes compared to people without? For example, asking whether people who are good at math tend to have different learning styles than people who struggle with it. So let's look at who exactly they studied and how they went about collecting their data. The details of how you conduct research really matter for understanding what the results mean. Their sample included 42 bipolar patients, 25 females and 17 males, and 40 matched controls with 25 females and 15 males. So they tried to match the groups on gender, which is good practice, like you making sure you compare apples to apples rather than apples to oranges. All participants were Croatian and recruited via opportunity sampling. This means they basically took whoever was available and willing to participate, rather than using random sampling. It's like surveying people at a shopping center. Convenient, but maybe not perfectly representative of everyone. The diagnosis was done via SADSL interview by 2 psychiatrists. SADSL stands for Schedule for Affective Disorders and Schizophrenia, lifetime version. Having 2 psychiatrists do the diagnosis helps ensure accuracy. It's like having 2 doctors independently confirm a medical diagnosis, much more reliable than just one opinion.
Quick pause. If you're finding value in this explanation, there's so much more to discover at Alt Academy, where we break down every A-level concept with the same clarity. Now back to the video lesson.
Now let's look at the actual genetic analysis. So how did they actually figure out what genetic variants people had? This is where the lab work comes in. They tested 2 genes. For the 5-HT2C gene, the alleles were called cysteine C and serine S. These are just names for the 2 different versions, like calling them version A and version B. For the 5-HTT gene, the alleles were simply called 1 and 2. Scientists aren't always creative with names. Remember, you inherit 2 copies of every gene, one from mom and one from dad. So for 5-HT2C, someone could have CC, CS, or SS combinations. For 5-HTT, they could have 11, 12, or 22. They took blood samples and analyzed them for these allele combinations. The lab work involves extracting DNA from blood cells and using techniques to identify which versions of each gene the person carries. It's like having a genetic fingerprint reader that can tell you exactly which versions of specific genes someone has. So after all this analysis, what did they actually find? The findings were actually quite interesting, even though they didn't find what they initially expected. Sometimes the most interesting results are the unexpected ones. Overall, there was no significant difference between the bipolar group and the control group. If you just looked at everyone together, you wouldn't see any clear genetic differences. It's like looking at a crowd from a distance. You might not see the patterns that become obvious when you look more closely. But when they looked specifically at females with bipolar disorder, they found some interesting patterns. This shows the importance of looking at subgroups rather than just overall results. Think about how certain medical conditions affect men and women differently. You need to look at each group separately to see the real patterns. Females with bipolar were more likely to carry the S allele of the 5-HT2C gene, particularly the CS type combination. This suggests there might be a gender-specific genetic vulnerability. They were also slightly more likely to carry allele 1 of the 5-HTT gene. Again, this was only in females, not in the overall sample. This reinforces the idea that genetic vulnerability might work differently in men and women. So what should we make of these findings? Let's evaluate the strengths and weaknesses of this study. Like any piece of research, this study has both strengths and weaknesses we need to consider. Understanding these helps us know how much confidence to place in the results. Look at the table in front of you. The strengths include rigorous diagnosis. Having 2 psychiatrists use a standardized interview means we can be confident that people really did have bipolar disorder. That's not always the case in genetic studies. They also replicated previous findings, which adds to scientific reliability. When different research teams find similar patterns, it makes us more confident the results are real rather than just chance. It's like having multiple witnesses telling the same story, but there are important weaknesses too, starting with the small sample size. With only 42 people in the bipolar group, it's hard to detect real genetic effects, especially when you start breaking it down by gender. All participants were Croatian, which limits generalizability. Genetic patterns can vary between different populations, so we can't assume these findings would apply to people from other ethnic backgrounds. It's like testing a new medicine on only one type of person. You can't be sure it'll work the same way for everyone. This study has some important broader significance beyond just its specific findings. Sometimes the implications of research extend far beyond what the researchers originally intended. It highlights sex differences in genetic vulnerability. The fact that they only found genetic associations in females suggests that men and women might have different genetic risk factors for bipolar disorder. This is really important for understanding how the condition develops and how it might be treated differently. It contributes to pharmacogenomics and personalized medicine. Understanding genetic differences could eventually help predict who might respond better to different medications. Like having a genetic manual that helps doctors choose the right treatment for each individual person. It supports the shift toward holistic models that consider both genes and environment. The fact that they found relatively weak genetic effects suggests that environment and gene-environment interactions are probably crucial. There are also some important ethical considerations we need to think about with this type of research. Genetic research raises some important ethical issues we need to consider. These are serious concerns that affect real people and families. Informed consent is critical because genetic information is permanent and could potentially affect family members too. Participants need to understand what they're agreeing to. It's like signing a contract. You need to know what you're getting into because you can't undo it later. There are privacy risks with genetic data. This information could potentially be misused by insurance companies or employers if it's not properly protected. Your genetic information is like having very personal information that could be used against you if it falls into the wrong hands. Importantly, we cannot predict risk with certainty due to gene-environment complexity. Even if someone has risky genes, it doesn't mean they'll definitely develop bipolar disorder. It gives you probabilities, not certainties. So let's see how it connects to what we'll study next. So let's wrap up what we've learned from this study and see where we're heading next. Oruc and colleagues' 1997 study is a key example of association analysis in psychiatric genetics. It shows you how researchers actually go about looking for genetic links to mental health conditions. They found no clear link overall, but discovered some important female-specific clues. This reminds us that genetic effects might be more complex and specific than we initially thought. Sometimes the most important discoveries come from the unexpected results. The study demonstrates the value of negative results in science. Even when you don't find what you're looking for, you learn something important about the complexity of the phenomenon you're studying. It's like Edison's famous quote about finding 10,000 ways that don't work. Each failure teaches you something valuable. Next, we're moving from biological explanations to psychological ones with Beck's cognitive theory of depression. We'll see how thinking patterns and cognitive processes might contribute to depression. It's a completely different approach to understanding the same condition. That concludes our exploration of the Oruc study. Remember, this shows us that while genetics clearly plays a role in mood disorders, the relationship is complex and probably involves interactions between multiple genes, gender, and environmental factors. The key takeaway is that genetic research is harder than it might seem. You need large samples, careful controls, and an understanding that genes don't work in isolation. But studies like this one are building blocks that help us understand the complex puzzle of mood disorders. See you in the next lesson where we'll shift from biology to psychology.
Hey there! If you found this A-level psychology lesson helpful, why don't you head on over to altacademy.org where you can sign up for the full A-level psychology course.
In addition to psychology, we offered 7 other A-level subjects. Thousands of students have already taken the leap. What are you waiting for? I want to see you there.